As we begin with our experiment, the first and the most important step is to arrange the workbench with all the necessary requirements such as reaction constituents and consumables like clean and sterilized micropipettes, tips, and mini coolers. Screen pause. 2. For handling RNA, one necessary preparation is that all your consumables should be RNase free, for which they can be treated with DEPC, diethyl pyrocarbonate, prior to use. One can perform qPCR through various methods involving RNA directly, or through cDNA. Here we have reverse transcribed our RNA to obtain cDNA. Screen pause over. 3. Once we are ready to go, I would like to draw your attention towards the most basic requirements for the preparation of reaction mixture required for a successful PCR run. It includes nuclease-free water, master mix. Here this is Promega master mix with cybergreen dye for fluorescence detection in cycle, forward and reverse primers for your target sequence, cDNA RNA of desired organism or tissue. Master mix are provided by different companies like Thermo Fisher, Promega, etc. It is important that you go through the protocol for the respective product and prepare for your reaction accordingly. Thaw each of these properly before use and place them back in mini cooler. 4. Then start by labeling your tubes to reduce the chances of error. Transfer your DNA to small 0.2 milliliter tubes, or generally called PCR tubes, and place them back in mini cooler. 5. With the following reaction, or the standard reaction mixture followed in your lab, add each of the constituents in a single 1.5 or 2 milliliters tube. We'll call this our reaction mixture.
Start.
Take 27 microliter of nucleus-free water.
Pause. वीडियो पोस्ट करना है?
No, no, hello.
After that, add 45 microliter of master mix.
हम pause कर लीजिए ना.
वीडियो?
हाँ.
Okay.
As we are preparing for 10 microliter in each well in triplicates, our total reaction mixture for single set of gene primers will be 30 microliter, which includes our DNA too. So accordingly, well calculate. It's better if you have good pipetting skills to proceed with any molecular work, because you are working with micro quantities, and most of the molecular reactions are time sensitive. Now, transfer the reaction mixture, which is 27 microliter to PCR tubes with 3 microliter of DNA, making it 30 microliter. Six. Tap for mixing it well and spin down the mixture through mini spin or short spin. Seven. Transfer 10 microliter of reaction mixture to each well in your pre-chilled PCR plate. Remember that while transferring there should be no error, and also keep a negative reaction at the end for checking the accuracy of your cycle. Use a sealer to seal your plate with. And after that you can also use plate centrifuge, or here you can also use downward tapping to bring everything on the bottom of each well. Machine. 8. Follow the standard protocol in thermocycler with the following steps, including denaturation, annealing, and extension, with the last stage being the rest stage of 4 degrees. So that your reaction is safe after completion of cycles in your absence. Here the most important thing to notice and one should be careful about is the correct temperature at annealing stage. You should have the knowledge of at what temperature your primers are binding accurately to your DNA RNA template. Screen pause. 9. The SYBR green used in the reaction binds with the double-stranded DNA in a minor groove with absorption and emission maxima at 497 and 520 nanometers. Screen pause over. Indication on screen of machine. 10. Denaturation is the first step of the polymerase chain reaction process. In this step, the double-stranded DNA template is heated to a high temperature, usually around 95 degrees Celsius, to separate the two strands of the DNA molecule. This is a crucial step as it breaks the hydrogen bonds between the nucleotide base pairs, resulting in the formation of single-stranded DNA templates that can be used for the subsequent amplification steps. 11. Moving on to the next step of the PCR process, which is the annealing step. In the annealing step, the temperature is lowered to allow the primers to bind or anneal to their complementary sequences on the separated DNA template strands. This is a critical step as it determines the specificity of the amplification. Hence, proper temperature is required as the primers are short DNA sequences that are designed to be complementary to the regions flanking the target sequence that we want to amplify. Screen pause. 12. Next, we move on to the extension step of the PCR process. In the extension step, the DNA polymerase attaches to the primer template and starts to incorporate complementary nucleotides. The temperature slightly rises to 65 to 75 degrees Celsius. DNA polymerase extends the sequence-specific primers with the incorporation of nucleotides. As the PCR primers are extended, more DNA becomes double-stranded. SYBR green dye binds to the minor groove of double-stranded DNA. The intensity of the fluorescence increases and is detected in the thermocycler. Screen pause over. 13. The final step for us is to decide the number of cycles required to get a substantial amount of amplicons required for the study. We generally keep this number from 35 to 38. 14. And then we'll start the reaction cycles and can visualize the melting curves and quantify the data through CQ or CT values. Thank you for joining and all the best.